Welcome to the presentation. I'm going to be covering the general chemistry relevant to introductory neuroscience. As a disclaimer, I'm not an expert in any of the affiliated fields, and this is going to be a layman's summation of the general basic tie-ins between general chemistry and introductory level neuroscience. With all of that said, let's get into the presentation. So starting with basic anatomy, the nervous system consists of all of the nerves in the human body. This is generally divided into two main areas known as the central nervous system, which is comprised of the brain and spinal cord, and the peripheral nervous system, which is literally everything else. That's going to be your sensory neurons, motor neurons, nerve plexuses, and interneurons in order to transport signals. So everything between the central and peripheral nervous system is generally wired contralaterally or to the opposite side, with the exception of hearing. Well, many exceptions, but hearing is the most pronounced exception. So any signal from the right side of the brain is going to lead to the left side of the body, and vice versa. This can be clearly seen in stroke patients, where a stroke on the right side generally leads to symptoms on the left side, such as left neglect or a contracture on the left side. All right, so starting with the central nervous system. The central nervous system is the primary processing center of the entire human body. This consists of the brain, which is the area in the head consisting of several specialized lobes and regions, with each lobe either providing an independent function or assisting in the coordination or interpretation of these functions. And the spinal cord is the other part of the central nervous system. This extends dorsally all the way down the back, surrounded by vertebrae. It's responsible for sending signals from the peripherals up to the brain and receiving those initial peripheral responses. Another thing that it does is causes immediate muscle interactions to knee-jerk stimuli. An example of this would be, the most common example is putting your hand on a hot stove, which causes you to clench away in pain, which actually works relatively counterintuitively when you're working with hot glassware, and is the reason that you're supposed to approach it with the back of your hand, because if you approach it with a large open hand, then your muscles naturally contract, which leads you to squeeze down on it, and squeezing down on hot glass is much worse than being in close proximity to hot glass. Another example of these immediate reactions is actually one of the benefits of Wing Chun Kung Fu. So Wing Chun Kung Fu, there's a lot of muscle memory involved in knowing what actions you can do, having the general quality of motion down in your brain. But additionally, those with enough training kind of naturally articulate their arms to facilitate the incoming force without really thinking about it. So neurons in the central nervous system don't regenerate, which is a major medical concern for the primary reason that damage like traumatic brain injuries, strokes, and spinal cord damage can result in long-term and lasting injuries and issues with that. So the peripheral nervous system, so motor neurons are a major part here. Motor neurons are neurons that send signals and make stuff happen. This is usually in the case of muscular contraction, though technically motor is a widespread term. When you hear most people referring to motor neurons, they're talking about muscle contraction neurons. And muscle contraction occurs via sending the neurotransmitters glutamate or acetylcholine to the muscles, causing the actin and myosin to shorten, thus contracting the muscle. So active contraction is how we do all of our mobility, and a very important process. Now, sensory neurons are also very important because it's what is necessary to perceive the entire world around us. They depolarize when faced with a certain stimulus, and based on this depolarization, they send signals up to the brain, which causes a response or processing of this stimulus, which is very useful in a variety of circumstances. Very few creatures lack, very few cellular entities lack, well, very few animals lack sensory neurons of some variety. Only the most primitive, like sponges, for instance. And there are several ways to classify these sensory neurons in humans, but the way that we're going to focus on throughout this presentation is modality, which is effectively how they function and what role they serve. Okay, interneurons are also found in the peripheral nervous system, and these are simply there to allow for interactions between neurons over a long distance, or in other words, to transmit signals from neurons to other neurons. And nerve plexuses are major structures. One prominent example of this is the brachial plexus located in your armpit. This is effectively like a junction box where everything comes together, and so everything can kind of be sorted by major system, and you don't have to have a bunch of disconnected nerve fibers, which would be much harder for the body to keep up. All right, epinephrine. So epinephrine is responsible for the sympathetic response, or the panic response. It's released by the adrenal glands on top of the kidneys and is responsible for revving your body up for dangerous circumstances. This means that it is what causes the release of non-vital muscles like the bladder and colon in emergency situations, as well as greater metabolism and a faster heart rate to facilitate greater movement of oxygen throughout the body in order to keep your cells oxygenated while they're metabolizing quickly. Additionally, it causes an increase in cognitive functions, so that you're more aware of the circumstances, which coincidentally increases memory. And just as a fun fact, snakes, similarly to spiders, are an evolutionarily ingrained fear in humans because being in proximity to snakes and spiders due to the venomous varieties can tend to have negative impacts on short-term health. All right, Schwann cells. So Schwann cells are a pretty interesting thing. They're additional cells that aren't nerve cells that allow for quicker movement of the electricity within cells. And the electricity isn't exactly like the electricity in a wire as we generally think of it, which I'll get into later. However, it does function similarly enough for Schwann cells to maintain all of the same functions as, let's say, insulation on a wire would. This means that it increases the resistance, which due to Ohm's law, dictating that resistance times current equals voltage, increases the voltage. Schwann cells, their membranes, as with all cellular membranes in the human body, consist of phospholipids. Phospholipids are completely non-metallic. Will come up later in this presentation, and as discussed in Chapter 4, them being completely non-metallic, non-metallic substances have a poor capacity to conduct heat and electricity. All right, so where does the charge in this system come from? It's ionic charge, which is moved around via, of course, differences in attraction of ionic charge, also diffusion, movement via that is an important thing, and that was briefly touched on in reference to gases in Chapter 11, and gated pathways, which allow for facilitated movement rather than constant movement towards the lowest potential, and neurotransmitters, ATP and stimuli, are what cause these pathways to open. So that will also come up later. All right, electrolytes. Electrolytes, as discussed in Chapter 9 of General Chemistry 1, are substances which completely iodize or dissociate in water or an aqueous solution. So major electrolytes in play in the nervous system are those phospholipids that I mentioned earlier. All of them are anionic, which means that they carry a negative charge, which results in the inside of cells having a negative charge. Additionally, sodium, potassium, and calcium cations, or positive ions, are important parts of this process. There is more potassium on the inside of cells and more sodium on the outside of cells. And this is important because of the function of the nervous system and differences in pathway function, as well as it's important in nutritional science because you lose more sodium via sweating and other physical activity than you lose potassium. And that leads to the example I have here of a comparison of the labels of Gatorade and Prime. Whereas you can see here, Gatorade has 270 milligrams of sodium and 80 milligrams of potassium per serving. And the serving sizes are a bit different, but once I go over to Prime, you're going to see the comparison there. So a good balance, more sodium than potassium. But as we look over at Prime, Prime only has 10 milligrams of sodium per serving, which it's a lower serving, so that's permissible as long as their potassium balance is fine. But the potassium balance is 700 milligrams of potassium. So that means that Prime has a greater amount of potassium than sodium, which means that it's replenishing electrolytes in improper proportions for high-performance athletic activity. So that's something to keep in mind when making consumer decisions in the future. All right, now for some practice questions. These practice questions are going to be based on the label provided. So find the molarity of sodium of 1 bottle of Gatorade. So 1 bottle of Gatorade is going to be 1 serving size. The conversion rate of liters to fluid ounces, which is what it's measured in there, is 1 liter for every 33.814 fluid ounces. And there are 270 milligrams of sodium in 1 bottle of Gatorade. So if you want to try and solve that, go ahead and pause the video. And the answer is 0.02 moles per liter, or molar. And you get this by taking 2 sig figs because the measure of fluid ounces is in 2 significant figures. First, you take the amount of sodium in grams, then you divide that by the molar mass of sodium to get moles, then divide that by the number of liters, which you get from a basic unit conversion. All right, next question. How many kilojoules of energy would you need to burn? Would you need to burn all of the calories in 1 bottle of Gatorade? So how many would you need to exert to burn all of those calories you just got from a bottle of Gatorade? And the conversion rate of calories to joules should be on the sheet that you were given with the periodic table. If you don't have that resource on hand, feel free to Google it. But anyways, we can see here that it has 140 calories, and that's capital C, which means it's kilocalories. So if you want to solve that, go ahead, take a few seconds and do that. Pause the video. And the answer is 586 kilojoules. It's basically a direct conversion because kilocalories to kilojoules is the exact same thing as a calories to joules conversion. All right, depolarization. This is the quintessential process of neurological function. It's what causes this communication between neurons. And differences in charge and diffusion are fundamentally what drive this process. So a stimulus gate on a neuron opens, which allows in sodium. After that sodium enters, if the stimulus gate is open long enough, it creates an action potential, which means that enough sodium got in to drive it up past that negative 55 millivolts necessity in order to drive an action potential. So if action potential is met, the sodium ions that just went in activate voltage gates at nodes of Ranvier, or however it's pronounced. I am not strongest individual with French phonetics, along the axon of the cell, and these nodes are effectively just between Schwann cells in the myelin sheath, or in unmyelinated cells just intermittently. And these opened voltage-gated sodium pathways allow more sodium to flow in, which then moves to the synapse area. At the synapse, it opens voltage-gated calcium channels. Calcium ions come in, they bind to the vesicles, which carry neurotransmitters. The neurotransmitters go down to the bottom in those vesicles and release into the synaptic cleft, where they then interact with other neurons, giving an inhibitory response, which decreases chance of depolarization, or an excitatory response, which increases response of potential for depolarization. All right. Now, repolarization, if this didn't occur, then neurons could go off only once, and then they would be completely useless for the rest of eternity. But repolarization, how that happens, the sodium gates close, which is a naturally occurring thing that I don't really have the time to go into in depth right now. Potassium gates open, and potassium diffuses out because there is comparatively less potassium on the outside than on the inside. And then to restore the original balance so it can repeat the process, it needs to activate the sodium-potassium pump, which uses ATP to trade 3 interior sodium cations for 2 exterior potassium cations in order to restore that balance. So the sodium-potassium pump works against the concentration gradient and against the natural difference of charge, and thus requires ATP in order to have the energy to actually undergo this process, because it's a process that requires energy inputs. And because of this ATP-intensive process, the brain actually uses 20% of all of the calories that your body uses, according to some studies. All right, now for a minor tangent about ATP. ATP is a nucleic acid. It has 3 phosphate groups on the end in a chain format. Phosphate group 3 is comparatively less stable because it's anion positioned directly adjacent to other anions, and so isn't very stable. The process that it causes it to break off, or the reaction, has a negative enthalpy of reaction. It's exothermic, so it generates energy when that thermal breaks off due to some mechanics that Dr. Resniak said we would get into in 1120. And this is important because the transition from ATP to ADP allows for the generation of the energy needed for basically all of the body. All of the cellular operations of entities that respirate. So, basic operations of psychoactive drugs. Disclaimer, do not take psychoactive drugs unless you are legally prescribed them by a state-licensed psychiatrist. Okay, now that I've got that out of the way, let's go into what they do and what they are. You've got your agonists and your antagonists. Agonists cause something to happen. Antagonists work against something happening. An example of an agonist would be tetrahydrocannabinol. Tetrahydrocannabinol binds to cannabinoid receptors, creating a variety of excitatory and inhibitory effects. Now, this ultimately results in damage to the hypothalamus or alters operation of hypothalamus. So those under the effects of tetrahydrocannabinol or who have been formerly exposed to large concentrations of it can actually have lower inhibitory responses and poor temperature sense because of the operations of the hypothalamus. And reuptake inhibitors, this is an interesting one. Drugs that fall under this label would be SSRIs, or selective serotonin reuptake inhibitors, and certain Freud-endorsed white powder that I'm not going to say by name. So how these work is they, after the vesicles release neurotransmitters, these things go in and they stop the reuptake process of these neurotransmitters. So you've just got neurotransmitters floating all around the synaptic cleft, continuing to do their operation because they're not being reuptake, because they are not receiving the reuptake and thus are still available for use. All right, now we're going to get into some of the modalities of sensory neurons. This is mechanoreceptors. So mechanoreceptors, you've got your audio receptors, your touch receptors on the skin, your baroreceptors, also known as your pressure receptors. So how these work is a physical stimulus causes a deformation in the membrane, which allows for channels to open and thus causes an action potential to be triggered. Now these can be encapsulated, which means they're wrapped in connective tissue, or unencapsulated, which means they aren't. These can be quickly adapting, meaning they just tell you when an impulse starts and stops, or slowly adapting, which means that they tell you and that they receive a constant signal, which actually allows you to tell shape and pressure of things. It's why when you hold dice in your hand, you can tell where all the bumps and ridges are based on your sense of touch, because it's constantly giving signals to your slowly adapting receptors. And light touch or heavy touch sensitive neurons, that distinction effectively just means heavy touches are going to trigger some, light touches are going to trigger others. And the receptive field actually depends on the area. So in areas in your back, a few millimeters apart can actually have the same receptive field, which means that if hypothetically two like really fine needles touched really close together, it would only feel like one touch response. Whereas receptive fields in the hand, for instance, and other areas like that are much closer together, so it requires a much shorter interval in order to actually result in that. All right, next up, chemoreceptors. These are pretty intuitive. So a chemical binds to the receptor, and then the receptor shoots off an action potential. And because the chemical incites an opening of the sodium ion channels. And some notable examples of this, there are going to be your pH and hormone receptors on the interior of your body, and of course your olfactory and taste receptors on the outside of your body. These are important for a lot of reasons. One is to make sure you don't poison yourself while you're eating or smelling stuff or being in different areas, because it's designed to associate bad smells and bad tastes, such as bitter, with things that are poisonous. All right, thermoreceptors. Thermoreceptors respond to temperature. Pretty straightforward. The membrane is susceptible to changing based on the temperature of the surroundings. Different types of neurons have different types of openings, and all of these openings and ion channels actually respond to different temperatures. So depending on what signal you're getting, it's going to tell you what you need to do in response to it. So if your internal temperature is off, then your body's going to do weird metabolic stuff. Whereas if your external temperature is off, it's going to send you signals that incentivize you go to a place with a better external temperature. All right, nociceptors. These are pain receptors, and they can actually behave in the modality of either mechano, chemo, or thermoreceptors, which means the modality of the three modalities we just touched on. So these can be myelinated type A delta or unmyelinated type C. A delta is that sharp pain, that initial pain, think like a needle when you're getting a flu shot, whereas type C is more of a sustained pressure pain, like the after effects of getting punched in the arm. Another example of exercise that you can do for this, don't try this unless you are willing to experience the affiliated pain, okay? Disclaimer. All right, so You're going to take your thumb and middle finger, position your arm in the standard anatomical position, then take that around the biggest point in the arm, that should be the left and right side, go up 1 to 2 millimeters, maybe a centimeter, and you should feel kind of drop off right there. Now if you squeeze on that, it should elicit a pain response, and that's mostly type C pain. You can move your muscles around, kind of accentuate it, feel how it feels at different angles, or not. I really would not recommend doing this drill or exercise independently. It hurts. All right, photoreceptors. Photoreceptors are rods and cones, and they work rather counterintuitively. So rods have rhodopsin and cones have iodopsin. But both of these are fundamentally an opsin protein tied to a retinol. All the retinels are the same, but the opsin proteins differ. And the opsin proteins differing is important because different opsin proteins respond to different wavelengths of light and absorb them better. So innately, there's cyclic guanine monophosphate, or CGMP, in these cells that keeps the ion gates open, creating the dark light response, which turns on rods but turns off cones. So whenever energy of the right wavelength is absorbed by the opsin, it gives the retinel enough energy to change from its cis isomer to its trans isomer. And this results in it dissociating from the opsin. The opsin then becomes an active opsin and breaks down the CGMP. And this turns on the light response, which results in rods turning off and cones turning on. Rods and cones send their signals to ganglion cells. Rods are relatively weaker, so they can sometimes even have hundreds of cone cells sending signals to one ganglion cell, whereas cones are much stronger, so they will have fewer, if not just one cone cell sending a signal to a ganglion cell. And then these signals are processed, sent contralaterally to the opposite side of the occipital lobe, which is then flipped upside down and backwards and then processed as an image. So it's a relatively convoluted process. There are more details, but that's more for a time talking specifically about optics and science regarding the eyes, a bit out of the scope of this presentation. All right, another practice question. How many sigma and pi bonds does this retinol molecule have? As a hint, because of the denotation on the transretinal, it may not be as apparent in that sample. Pause if you want to try and find the solution yourself. And the answer is 22 sigma bonds and 6 pi bonds. And really the important part about that is this pi bond right here, that is what results in it needing an energy transfer in order to change from the cis-retinal to the trans-retinal, and as a result, why it's capable of responding to light. Anyways, sources, a video source, and I will post all of the sources or links to them anywhere that I post this. That's all for me, so goodbye, and thanks for watching.